assayed for levels of inflammation. Then they underwent the MRI scan using that task that I mentioned to you previously, and then we took 3 blood samples from them following the scan, which were again assayed for levels of the pro-inflammatory cytokines, and ultimately they were dismissed. So specifically, we were interested in exploring how the brain responded to negative feedback, such as being called annoying, compared to neutral feedback, such as being called sensible, given that we thought that it was really responses to this negative information that might be driving the inflammatory response. So just a quick manipulation check to show you that this was indeed a negative stressful experience. You can see participants show significant increases in negative affect and feelings of social rejection from before to after the social evaluation stressor. And just some qualitative data to give you a better sense for what this is like. We had one participant who asked the nurse who had come in to take a blood sample from her, "Do I seem annoying to you?" after the scan. We had another participant who told us upon being debriefed that she had planned to call her roommate as soon as she left the scanner and ask if she came across as insecure. So definitely an experience that, although they're just lying there in the scanner getting this feedback, is stressful and distressing. But we weren't sure if it would be enough to elicit an inflammatory response, so that was the first thing we wanted to check. So what you'll see here is time on the x-axis and levels of one measure of inflammation, the pro-inflammatory cytokine interleukin-6, which is on the y-axis. And as you can see, we do see significant increases in levels of inflammation at essentially every time point following the stressor. So participants did show an inflammatory response to undergoing this experience of social evaluation. Next, we were interested in sort of just ignoring the inflammation data for the time being and just looking at what was happening in the brain when they were receiving negative feedback compared to receiving neutral feedback. And what you can see here is that participants showed greater neural activation in mentalizing related regions, so these regions that are really involved in understanding what other people are thinking and feeling in response to specifically the negative information. So this kind of fits with the sort of bad is stronger than good idea, that when someone's giving you negative feedback, you're sort of really bringing on this mentalizing system to try to potentially understand why they're thinking about that, why they're thinking that about you. But the main goal of the study, again, was to bring the neural data together with the inflammatory data and to look at what in the brain during a negative social experience would be associated with a stronger inflammatory response. So that's what I'll present to you here. Essentially, what we did in this analysis was compare neural responses to negative feedback and searched across the whole brain for regions whose activation was correlated with the inflammatory response. Again, to try to get a sense for what was in the brain that was active during the time of the negative feedback was associated with this downstream increase in inflammation. And what we found was that the amygdala, this key node of the threat-related neural network, was positively correlated with inflammatory responding. So here are the same data shown here with amygdala activity on the x-axis and inflammatory response on the y-axis, and you can see this positive relationship between amygdala activation to negative feedback and increases in inflammation. So again, greater amygdala activity to greater negative social information is related to stronger inflammatory responses to social evaluation. So this was interesting. This was the first study to show a neural underpinning of inflammatory responses to social stress. But as I mentioned before, we were really interested in trying to figure out how this threat-related neural system might be working in concert with a mentalizing-related neural system to really drive inflammatory responses. So we did some functional connectivity analyses to try to look at this. So I just want to walk you through what we're doing in this type of analysis because it's a little bit complicated. So the analysis we did is called the psychophysiological interaction, or PPI. And what we did was we divided our sample into high and low inflammatory responders just based on a median split, which actually did a pretty good job in this case of representing people who really didn't show that much of a change in inflammation compared to those who showed a stronger inflammatory response. And this is just an example of what we might expect to see. So the red line here is the amygdala activation. The orange line is DMPFC, core region of the mentalizing network. And what you can see shown here is this is sort of the time course that we would expect to see what's happening in this region. And for low IL-6 responders, we would expect very little amygdala activity. That's kind of what that correlation that I showed you before was telling us. And really little concordance between DMPFC activation and amygdala activity. But for high inflammatory responders, we might expect to see something more like this, where during neutral feedback, activity in these regions is generally low and not particularly related. But then during negative feedback, you see elevations in both amygdala and DMPFC activity, and you can see that they're sort of turning on and turning off together. So activity in these two regions is correlated with negative feedback for high IL-6 responders. At least that's what we expected, again, based on this body of animal research that has suggested something like this might be going on. So looking at the actual data, we looked at neural activation in the amygdala, shown here in blue, and searched across the whole brain for other regions whose activity followed the same pattern and the same time course of activation as the amygdala did. And again, we were comparing those high IL-6 inflammatory responders to low. And what we found was that there was greater coupling of, or functional connectivity between the amygdala and the DMPFC, this core mentalizing region, during negative feedback, specifically for the high inflammatory responders. So this kind of suggests the possibility that it's the degree to which these two systems are really working together during negative social information processing that's driving this increase in inflammation. So to sum up from this study, we learned that activity in the amygdala during negative social feedback is related to inflammatory responses to social stress. We also learned that heightened functional connectivity, or this greater coupling between amygdala and DMPFC, or threat and mentalizing systems, was related to high IL-6 responding. And again, this is suggestive of a aversive amplification circuit, whereby you have this kind of initial threat response, and then these other potentially more social cognitions kind of come into play later on and then potentially drive greater threat responding. However, we need to experimentally manipulate mentalizing processes to examine their causal role in this circuit. And that's a study that I'd love to do, where we could potentially direct people to either really pay attention to these evaluators, try to figure out what they're thinking or how they're feeling, versus in another condition either do some sort of divided attention task or have people tell them to kind of ignore the evaluators and see if that changes the relationship between activation in these regions and ultimately inflammatory responding. So again, before I move on, I'll just sum up with some current and future directions in this domain. First and foremost, I really want to bring the social context back into this work. So one of the first studies that I'd love to do as a faculty member is, my first grant that I would love to write is really one that explores this entire circuit. So again, measures of and recruiting individuals from different socioeconomic or social status levels and seeing how that modulates their neural responses to negative feedback in ways that can alter inflammatory responding. And we did, as I mentioned before, take the latter measure in the study, and we do find some evidence that it's really this DMPFC region, that core mentalizing region, that seems to be driving inflammatory responding for lower status individuals. So we've run some mediational analyses where we're looking at status predicting levels of inflammation mediated by activation in that brain region. They're a little underpowered, so we're finding some marginal effects at this point, but I think it's at least pilot data for a grant to suggest that that's what could possibly be going on here, that in these threatening contexts, this tendency of lower status individuals to think about the thoughts and feelings of others may be having these health consequences later on. I'm also interested in extending this to look at social evaluation in an intergroup context. And this is a study that I'm working on currently with my mentor at UCSF, Wendy Berry-Mendez, where we're exploring the neurocognitive mechanisms underlying attributional ambiguity, which, as you probably know, is this idea that when individuals from minority groups get positive feedback from individuals from majority groups, that actually might be more distressing or threatening than receiving. And the idea is that there's sort of this attributional ambiguity about the positive feedback. Is this person saying that they like me because they actually like me, or are they saying that because they don't want to appear prejudiced, or they're trying to sort of impression manage and not appear to have these biases against me? So the way that we're doing this is recruiting a sample of African American participants who send in a picture of themselves, and we're telling them that we're having a number of other people rate their picture and what they think that they would be like based on their picture. And then during the scan, we're showing them feedback such as this. So this is a trial where the participant is getting positive feedback. You seem very kind, from an out-group member, a white person. And we're contrasting what's happening during trials like this, when they're getting positive feedback, like you seem caring, from an in-group member. And I think this could potentially help address an underlying question in the attributional ambiguity literature, which is that it could be potentially, for evolutionary reasons, that it's just less rewarding to get positive feedback from an out-group member. And we could see just sort of lower levels of neural activity in reward-related regions to this positive feedback from an out-group member. Or it could be that initial responding, these two types of feedback activate equivalent reward-related responses. But then some other mechanisms kick in that say, oh, I don't know if I can trust this feedback. I don't know if this person really thinks I'm kind, or they're just trying not to appear prejudiced, which might be reflected in sort of activation in these other prefrontal brain structures. So I think a good use of imaging technology to try to tease apart these two different processes that could be contributing to the distressing nature of this type of feedback. Okay, so in the last few minutes of my talk, I want to turn to the third area of my work, which has explored how the physiological state of the body feeds back to the brain to alter perceptions of the social world and our neural responses to social information. So going back to my diagram here, I've been talking about all of this as though, social hierarchies influence the way we respond to social interactions, which in turn influences levels of inflammation. But interestingly, these arrows also go the other way, and levels of inflammation and other physiological responses in the body can feed back to the brain to alter our perceptions of the social environment and potentially alter our perceptions of where we stand in a social hierarchy. And there's specifically reasons to believe that inflammation may be play a key role in this process. So in addition to what I mentioned before, that social environmental factors can influence levels of inflammation, increases in inflammation have also been shown to influence our affective experiences and our social behavior. So increases in inflammation induced experimentally, or in the case of these chronic disease conditions that I mentioned before, are associated with increases in negative affect and heightened depressive symptoms. And interestingly, also social withdrawal, where individuals in an elevated inflammatory state tend to be more socially distant and not as interested in or rewarded by social experiences. And the idea here is, if you're sick or injured, it might make good sense to withdraw from your social environment so that you don't infect other people, or so that you put yourself at less risk for experiencing an additional insult. But again, in our modern world, if we're kind of constantly mounting these inflammatory responses, this could potentially be a negative thing. So to address this issue, we ran a study where we randomly assigned participants to receive endotoxin to induce inflammation or placebo. Endotoxin is actually a piece of the cell wall of the E. coli bacteria that we inject into participants that mounts this. It sounds scary, but it's not. It makes them feel sort of, I would say, like if you've ever felt a little bit sick and under the weather, but not bad enough to stay at home in bed. So you're still kind of up walking around, but you feel like a little bit off. It's safe, it's time limited, but it causes levels of inflammation to go way up to try to fight off this pathogen. And we scan these individuals using fMRI when their levels of inflammation were at peak. So it's a model of what might be going on in something like depression or chronic disease. And they underwent that same social evaluation stressor used in the prior study that I mentioned, where they're getting positive, negative, and neutral feedback from an evaluator. So just quickly to show you that this manipulation is extremely effective in increasing levels of inflammation, you can see the red line here that the endotoxin participants are showing a huge inflammatory response, and we scan them right here when the inflammatory response is at its peak. And what we found when we looked at what was happening in the brain during negative social feedback compared to neutral social feedback was that there was greater neural responses in the amygdala, shown here, this key threat-related neural region, greater for the endotoxin participants compared to the placebo participants. So just to show you these same data again, you can see here that the endotoxin participants who are in an elevated inflammatory state are showing much greater threat-related responses to this negative social evaluation compared to the placebos. And interestingly, if you look just within participants in the endotoxin condition, so only people who are experiencing this heightened inflammatory state, and you look at how neural responses in the threat-related region are associated with their degree of inflammatory response, you see a positive relationship. So activity, greater activity in the amygdala in response to negative feedback associated with a greater change in inflammation. So these data suggest that acute increases in inflammation are associated with greater amygdala activity to negative social evaluation, which may reflect sort of greater threat perception or increased salience of negative information when we're in this heightened inflammatory sort of sick state. We also found that individuals who showed a stronger inflammatory response to this endotoxin challenge had greater amygdala activation to the negative evaluation. So it's not just the between group differences, but also within this sick group, the extent to which you show an increase in inflammation is related to neural responding. So just to wrap up, some current and future directions in this area. I'm currently analyzing these data to look at how inflammation affects neural and psychological responses to positive feedback. So my hypothesis is that when you're in this heightened inflammatory state, you might also be less sensitive to positive feedback as well. So it's sort of the negative is more negative and the positive is less positive. I'm also interested in exploring how blocking sympathetic nervous system activity might affect social perception. So sympathetic nervous system is thought to drive these inflammatory responses. So you get initial sympathetic activity and that then induces an increase in inflammation. And I'm curious if we sort of knock out that sympathetic response using drugs like beta blockers, if this can break the link between our perceptions of evaluation and increases of inflammation. And I think it'll be really interesting to explore if people have similar neural activity and sort of feel the same way in response to getting negative feedback, but they just don't mount that initial sympathetic. that changes how they feel. So when we're stressed, is it really, the beating heart and the sweaty palms that's cueing us that, oh, we must, be stressed out or feel distressed? Or is it that even if you remove that sort of physiological response, people will still think, oh, this person is calling me names and I don't feel good about it, but they just won't be able to mount an inflammatory response. So that's my body of work to date, exploring how the social context and social hierarchies influence our neural responses to social information and social threat, and how that links with downstream physiological processes that can be related to health, and then also on the way back up, how physiological changes influence the way that we respond to the social world. I'd just like to wrap up by thanking my many collaborators, especially my fantastic 3 mentors, Naomi Eisenberger, Wendy Berry-Mendez, and Dacher Keltner, my labs at UCLA and UCSF, and the funding agencies who have supported both me and this work. And thank you all for inviting me and for listening.
I just have a question about your participants. Were they all women, mixed gender? Did you see any differences in either sensitivity to social information or self-reported status levels, anything along those lines?
Great question. So in the 2 status studies that I mentioned, we didn't restrict based on gender, and I didn't see any relationships either in terms of gender differences in their self-reports of their status, or that gender sort of moderated the relationship between status and neural activation. In the second study, we recruited exclusively women, partially because we sort of know from prior work that women tend to be more sensitive to social stress and social evaluation, and actually are at greater risk for some of these chronic diseases, especially things like depression and rheumatoid arthritis. So it seemed to make both theoretical sense and sort of practical sense to focus on them. So we didn't look at gender differences in that study because we only had one gender. Although I think that would be really interesting to look at in the future. And also I think, now that we've sort of established that this paradigm is capable of eliciting an inflammatory response that sort of has the effects that we expect it to, it will be really interesting to play with the dynamics. So what happens when you change the evaluator? So all the evaluators were also women, so gender match. What if the evaluator's a man and the participant's a female? What about the reverse? What if, again, the evaluator is an outgroup member? So I think there are a lot of ways that we can sort of use this task in different contexts to ask different, but I think also important questions.
Is the evaluator always matched to the participant's race?
No, great.
Yeah,
it's a great question. So we did not, we sort of let it naturally vary. You could probably see we had a pretty diverse sample, kind of reflecting the UCLA student population, and our RAs were pretty diverse as well in terms of racial background. I did look at if there were any differences in sort of if they happened to be matched versus mismatched. Exactly, but with 30 people, it was such a small group that I actually didn't even see any differences, which is not necessarily what I would expect if I had recruited for that particular reason and looked at that issue. So no differences in this sample, depending upon if the confederate or the evaluator was racially matched or not. But again, that's sort of something that I'm really interested in exploring and kind of building on in my postdoc work. Yeah?
So throughout your talk, I was trying to think about what the mechanisms would be. So initially, I was thinking, well, with social status, that people who are lower in social status, for example, those who've been bullied or for whatever reason, are going to attend more to threat and think about others because they have to figure out what other people are doing.
Right.
And then I was thinking, with the later studies where you induced, where you injected people with neurotoxin.
Endotoxin. Endotoxin.
Yeah, boy, I've got to be careful with the language. I mean, if I feel under the weather, I might also feel a little bit depressed. So
Yeah.
was mood coming into play?
Right.
There was connection between mood and
Absolutely,
inflammation.
absolutely.
So what are your thoughts about the mediators? And I'm sure there are multiple ones.
Yeah, it's a great question. I think I've been thinking about the first set of data in the same way that you do, that it's sort of necessary and adaptive for a lower status person to be kind of scanning the environment and really, trying to figure out what other people are thinking or feeling, and, does this person mean to do me harm or not? So I've been thinking about it that way as well. In terms of the inflammation study and the endotoxin data, yes, I think that mood is probably definitely playing a role here. And one of the things that I'm trying to look at now is if neural responses to this negative feedback actually are associated with changes in mood. So sort of in a mediational model, does inflammation versus not predict changes in depression? We know that does. And is that mediated via activity in this threat-related region in response to negative feedback? Yeah, so I think that's exactly right. And those are the types of things that I'm currently looking at. I'm also interested in, you mentioned mood, but what about status? Sort of bringing it all back together. Does being in a heightened inflammatory state lead you to feel lower status? And sort of one of the things about this SES and health relationship that no one really talks about that much is that most of that work is correlational. And it could be that, we know, for example, that being sick obviously can drive decreases in SES, for example. And I think we know less about how it changes subjective status. So I will say preliminarily that it does seem like there's a relationship where we had people rate themselves on the ladder before getting this injection, and then when their levels of inflammation are at their peak, and there are changes where people do report feeling lower status when they're in a heightened inflammatory state. So that could, I think, be another. I guess you were thinking about it in terms of mechanisms. I was thinking outcome, that the neural data could be a mechanism toward explaining. But yes, absolutely something I think to look at. Yeah?
So speaking of correlations, the first couple of studies that you presented were essentially correlations.
They were.
You looked at a relation between social status, perceived social status,
Mm-hmm.
utilizing perceived social status, and threat perceptions, and then mentalizing threat perceptions.
Right.
And situation,
Right.
So of course, the correlational data is only in the last third that you started manipulating.
Yeah.
Very good. So a couple of questions that occurred to me while I was listening to your talk, especially the first two-thirds. One is, what if you manipulated social status?
Mm-hmm.
Would you find differences in the relation? Is it the manipulate? Is it the objective social status?
Right.
Or the perceived? The second is, I would imagine that feedback from the other person would matter, especially if they're higher in social status rather than lower.
Mm-hmm.
Right? So I'm thinking of the connection between your work and previous research on power.
Mm-hmm.
That low power people are particularly sensitive to the emotions and the intentions of high power people,
Right.
not of others in their own status. So in your studies, have you either measured or manipulated the perceived power of the feedback giver?
Right. Yeah, the short answer is no. I haven't measured power. We've sort of created a power dynamic in this task, right, where one person is evaluating the other and giving feedback, so that sort of inherently affords them power, I suppose, to some degree. So, and that's true that could be sort of why the status level of the individual is predicting greater mentalizing. It would be interesting, although I don't know exactly how you would do this, to have the person be of similar, who's evaluating you, be of similar status. Sort of how do you create an evaluative environment that's not differential in status or power, but that would be interesting and would kind of tease apart that. Is it that lower status people just sort of walk around mentalizing more about everyone, or is it specifically to individuals who are higher power or status? With regard to the first question about you were asking about correlations and manipulating status. I have not done that. There's some work to suggest that's manipulating at least subjective perceptions of status by having people sort of focus on individuals higher than them in a hierarchy versus lower does produce the same types of effects. So that there have been studies that have manipulated people to feel higher versus lower power or status and shown similar effects. So I don't know if you guys are familiar with the study from a little while ago that Adam Galinsky did, where he had this creative, they had people draw E's on their foreheads, and sort of if you drew the E as though it would be like readable to someone in your environment versus that made sense for you, and they found that people who were induced to feel high power tended to write the E so it made sense to them, and lower power people wrote it so that someone in the environment could perceive it. So I think those types of data at least start to suggest some causal evidence, but it would be interesting to push it in the other direction too. What if I made you mentalize more? Would you feel lower status? If I say, you know, I'm really motivated to understand what everyone in this room is thinking and feeling right now, and maybe that's causing me to also feel lower status. So it could go the other way as well. And then lastly, I'll just say, I think the objective-subjective question is really important and not one that we have yet done well at teasing apart. And I would say, if anything, at this point, my data suggests more similarities than differences. So whether we measure objective or subjective, you know, we did that in those two studies where we had, you know, one study was subjective measures, one was more objective SES indicators, and we see at least, you know, to some degree converging evidence that both of those are associated with, you know, lower status or effective SES is associated with heightened mental. And I also think the cross diagonals are interesting. So what happens when you're low SES but high status? Like you happen to have a high status position in your community. You're the deacon of the church in a low SES African American community. So technically you have sort of, you know, lower SES resources, but you have greater social status. Yeah, absolutely. Right, exactly. And I think those are all interesting questions and things that I hope to follow up on. Sure. Yeah.
I'm just, you know, I have so many, I'm glad I'm going to get them. But one thing I was curious about, I know at the third section of your talk, we talked about positive feedback. But you did have, you know, a great deal of data on people's response to positive feedback.
Yeah.
And I'm wondering whether it actually mirrored, or whether there was simply, I mean, I have my own personal hypothesis about it, but
yeah.
One could imagine greater mentalizing, but no threat. So what
right.
happened with the positive feedback?
That's a good.
Is there
I have that slide somewhere here. The short answer is that positive feedback looks like negative feedback in some ways and not in others. So people don't mentalize as much, or you don't see as much mentalizing related activity to positive, but you see this sort of shift to these neural regions that are more ventral, sort of toward the front.